Okay, it looks like it's time for the last lecture. A sad occasion in no one's heart. So we were talking about the regulation of eukaryotic transcription. And I noticed that two of the DNA binding domains that are supposed to be there were not in the PowerPoint that I had in my pocket. So I downloaded the one from the website just a few minutes ago, and it has the two that come before the zinc-containing DNA binding domains, and that's the one you have, so we need to cover those. They're not real hard ones. You know, none of it's real hard. It's just way too much stuff, right? Who's in charge of that? I don't know. It's a wacky world. Okay, DNA binding domains. This is where it should have started. And we should have started with the very basic one here, the helix turn helix. And this thing doesn't work. There it goes. This is the one that goes all the way back to eubacteria. You know, it sounds like it should be 2 helices, but it's really 3 helices. So you have helix, turn, helix, but notice there's a structural helix in the background before you get to the first helix of helix, turn, helix. And also there is a anti-parallel, what's it called, beta sheet, 2 anti-parallel strands. So sometimes, these are not always there, but most of the time you'll have this helix turn helix supported, sort of coordinated and held in position by these other structures. So if you're looking on a multiple choice test, this is the text way to see this picture. Triple helical cluster reinforced with beta strands. That uniquely fits the helix turn helix motif. This helix here, it's in the major groove. It's called the reader helix. The other helix and this turn area make contact with the phosphodiester backbone. So the base specific contacts are embedded in this helix. And of course, it is not opening the DNA to read the bases. It is able to read the bases by looking at the edge of the DNA. It knows what the edge of an AT pair looks like. So that's kind of cool. So helix, turn, helix. Be able to recognize this structure. Here you see it being used for some of the genes they're called homeotic genes involved in development. We have homeotic genes, insects do. This is an example where one of the homeotic genes called engrailed has been mutated, and you can see the developmental mischief that brings about. Here's the head, and got its legs coming out of its head. That is so awkward, with the legs coming out of the head. Antennapedia. You got feet where your antennae should be. Homeotic genes, helix, turn, helix. It's an example of activators control development from insects to plants. Okay, the next is like something you'd see at the Louvre. A sophisticated art gallery in New York City. Imagine this made of gleaming silver. These 2 graceful helices coiled around each other. Simplicity in design and function. 1 helix serving both to recognize the DNA and to hold the 2 monomers together to form a dimer. All of that potential combined in 2 graceful helices. At the bottom part, the helix is basic because DNA is negative. That's where the base recognition event occurs. Once again, it's a helix sitting in the major groove. It's a dimer, so it's simultaneously reading both sides of the binding site. They come together at the top part as a leucine zipper. They interface, coil around each other because of this narrow strip that's 2 amino acids wide of hydrophobicity, which is how they stick together. So this is called a basic for this DNA binding portion, leucine zipper for the other part of the same helix that's required for forming the dimer. An example of it is our old friend here, GCN4. Key term that's not there yet, I'm going to have to put there. GCN4 is a transactivator from yeast. It's the one that is specific for SAGA. It cannot recruit TF2D. It's specifically there for SAGA. GCN4, beautiful B-zip. And here you see a more complicated sort of modern art version of the B-zip. This is fractured and then put back together. You see the graceful helices, and they get ready to twine around each other in oligomerization. And yet, there's a break. There's a loop that destroys the continuity, the flow. But it has its own beauty. It's kind of like two handles. You could pick that up and play it like a musical instrument. You could signal like a big ram's horn across the valleys. The helix, loop helix. Again, at the top part, you have the leucine zipper. At the bottom part, you have a basic helix. So you see, the fundamental design feature in many DNA binding domains is simply a protein helix. The protein helix is fairly rigid and can only interact with 1/2 of the DNA molecule. And because it takes 10 nucleotides to go all the way around, it's limited to being able to recognize 4 to 6 nucleotides at a time. You see the helix doing that same type of recognition in the zinc fingers and the 3 different forms of the zinc-containing DNA binding domains also. What's an example of this? MyoD is a transactivator in eukaryotes. It's involved in development of muscle tissue, MyoD. And this is where the slides took up last time. I haven't quite figured out why there should be some missing, but this is what we've talked about, the 3 kinds of zinc-containing DNA binding domains. Here you see a famous one here, Gal4. It's used in many constructs. It's used in the genome engineering project that you guys are doing. We talked about the regular zinc finger, the fact that the Gal4 contains actually 2 zincs. So when you're going through the DNA binding domain drill, just look for 2 metal ions, and the only thing that has 2 per monomer would be this bimetal thiolate cluster. And then we just begin to get into the nuclear receptors, that these are a class that have really 2 ways to use a zinc-containing protein motif. One is engineered, it's very similar to a zinc finger, and the other is used for forming the oligomer instead of a leucine zipper. This is a very distinct feature of the nuclear receptors, sometimes called steroid receptors, and in the fact that there are 2 modules, as they're called. So this is the regular zinc finger that we talked about. Here you see again a simple helix, but it's in conjunction with an anti-parallel beta strand, and there are 4 places to stabilize and make a coordinated connection by the zinc. Notice here we talked about a polydactyl zinc finger, meaning the zinc finger group of proteins have solved the problem of sequence complexity. Instead of forming a dimer to make the simple binding site that one helix can recognize more complicated so it doesn't appear by accident and you lose control of regulation, it has discovered that all you need to do is line these simple zinc fingers together in a single protein. And there's one called Krupel that has at least 13 of these in a row. So you certainly don't need a dimer of that thing. A monomer of something that's a polydactyl zinc finger, as you see, could be quite useful. You could really build in a lot of binding specificity with that sort of thing. So on the exam and in that question bank of structures, there's a polydactyl zinc finger. So look very carefully. If it has a zinc ion, you'll see a metal, and then you start seeing multiple things that have zinc ions, and they're all in a row. That can be only one thing, a polydactyl zinc finger. And here you see the actual details of the specificity of the interaction. These particular regions of the helix are recognizing the bases on one strand with some recognition on the opposite strand. So you see a 1, 2, 3, 4 base recognition motif, and there's some ambiguity here. Every zinc finger usually has an alternate binding too, but if you put 2 or 3 together, the overwhelming direction of binding would be towards the most probable binding site.
Dr. Gurley?
Yep.
How do the zinc fingers interact with one another?
They're not interacting with each other. They are kind of free floating, like marbles on a string, or I hate to say beads on a string. It seems like people could think of some other way to say beads on a string. But these are little independent, to me they look like little baby gators. They're all baby gators on a string as they go across. So they become rigid and fixed as they bind the DNA, and they would go wrap around the DNA so that you'd have these staggered recognition sites. But how they get folded, there must be some chaperoning involved in scanning and approaching their target. They probably randomly, a DNA binding domain will randomly make contact, and if it fits, then the next will try to fit. If it doesn't fit, then it's not very tight binding. It just goes someplace else until they all begin to click into place, and there you have a stable association. Okay, the nuclear receptor, and we talked about this. I think this is about where we stopped. Here you see the 2 zinc modules. There's 1 zinc ion here, 1 zinc ion there, and this represents a monomer. Each of the circled areas with the arrow represents a module. This is a monomer, and that's a zinc module within the monomer. These together make a dimer. Glucocorticoid is one well-characterized example. These have, I said it's like a zinc finger. Remember the zinc finger had 2 histidines and then 2 cysteines, and the bimetal thiolate cluster had 6 cysteines. Well, this is the uniqueness here of the zinc module involved in DNA binding. With the steroid receptors, it has 4 cysteines, no histines. You're not going to need to know that level of detail, just letting you know that that's where the difference occurs. So here's another aspect. They are not called receptors for nothing. They recognize the hormone. They directly bind the hormone, and we talked about how steroid hormones are membrane soluble. They can go right through the membrane. You don't need an embedded receptor there in the membrane facing outward. You can bind the transcription factor directly. Here you see more detail on how the 2 monomers, looking at their 2 respective zinc finger-like regions or zinc modules. You see how it's the same, it's just almost identical to the zinc finger. These are slides that cover how you get specificity here, and the spacing between sequence specificity. Let's see here. DNA binding, data sheet. in this region here, between the two contact points with the zinc. So, residues between the two His residues. I was wrong. The His residues, I meant to say, if I could read the slide. I know you're not supposed to read the slide except when you can't remember the slide. It's better than making up stuff. Between the two His residues, as I meant to say, that's where DNA sequence recognition is determined in the steroid receptors. Spacing between promoter elements. We'll see more about that in a minute, how that affects it. Okay, let's see. So that's the DNA binding motifs. There's a DNA binding motif drill there as a homework. Go through those so you can at a glance tell what it is. Because I'll take one of those on the exam and you'll have to tell me what it is. I don't know what it's going to be. It's going to be a random draw. So the next part of a transactivator is besides the nuclear localization motif, is the transcriptional activation motif. I mean, how complicated should that be to activate transcription? It sounds like it should really be something quite elaborate. It turns out, again, it's a very simple design. Whoa, man. So it's actually a couple of helices again. These helices have a very distinct distribution of charge and hydrophobicity. One side of the helix is charged, and the other half, the bottom half in this case, is full of bulky hydrophobic residues and is hydrophobic. And so that kind of distribution of charge and hydrophobicity on a helix is called amphipathic. This is an amphipathic helix. Amphipathic helix. This is a diagram of one that's really, really strong. This is in the one that causes the fever blister and other kinds of herpes. The herpes activator is very strong. A lot of virus activators are really, really strong. VP16, VP standing for viral protein 16, turns out to be just this activator. It doesn't have a DNA binding domain, so in a sense this would be a co-activator. It's just the activation domain itself as a free floating peptide. This thing is so strong it works like crazy in everything you put it in, even plants. Since plants don't have lips, they don't get fever blisters. But it's quite useful in genome engineering if you want to have a really robust expression of something. Amphipathic helix. And they found that it doesn't have to necessarily be negative charge. Even positive charge will work. So you can sort of envision how this might work. Turns out these helices are not real stable as a protein alpha helix. And they are kind of flexible until they actually bind to their target site, and then they assume a helical structure. What does that resemble? The hinge helix, remember, of the lambda repressor, LAC repressor. The hinge helix, when it makes contact with the minor groove, becomes stabilized and actually forms a helix. These acidic activation domains have that same kind of property. They're pretty flexible until they actually come in contact with their target. What would be a target? Anything that a transactivator would recruit. We don't know much about the target sites, how to build one of those, but we do know how to build a very strong activation domain, build it into a transactivator. There are several ways to build an activation domain into a transactivator. This is the easiest to understand. because the others seem like there's something crazy. We don't know. It's a stretch of alanines or glutamines or proline, serine rich. These are usually things that don't have rigid structure, and this principle of assuming a structure when they smack onto their target seems to be a common theme. But the other stretches of protein sequence that perform this role in other transactivators, how they attract, or how the basis of their recruitment or affinity is much less understood. So this is kind of the workhorse way that you build an activator domain. This would be coupled with a DNA binding domain, and you just built yourself a transactivator. In yeast, this is almost 100% the only kind of activation domain you find. Most other eukaryotes are more complicated. They have the variations of different kinds of activation motifs. Okay, so let's look at a specific pathway of gene activation in us. The steroid hormone pathways. How does it work? And what you can say for one is a pattern for what you can say for all. The specific details vary, but usually vary by degree, not in terms of mechanism. So here's a diagram of the glucocorticoid receptor. It was the first to be well characterized. You have, in this case, the DNA binding domain is sitting right in the center. And of course, you remember the one with 2 zinc modules, it's there. And then on the N-terminus, you have these acidic activation domains. This is where you recruit basal transcription factors and chromatin remodelers. And the other part of the protein, the C-terminal half of the protein, is for regulation. This is where the glucocorticoid hormone itself will bind. And it's where another protein called heat shock protein 90, HSP90, because they thought it was a part of the heat shock response. It participates in it, but it does so many other things that probably its primary role is with this kind of regulation, where it will mask a domain, and then when a ligand binds, it loses affinity, drifts away, and exposes something. In this case, it exposes the nuclear localization domain of the glucocorticoid receptor, and now the glucocorticoid receptor with the ligand, with glucocorticoid still bound to it, goes to the nucleus, finds its promoter, and activates a whole series of genes. So this kind of regulation, here you see the glucocorticoid on the outside of the cell, freely diffusing through the membrane, and then making contact with the glucocorticoid receptor, HSP90 leaving, and it going to the nucleus. This is called nuclear localization as a mechanism. The reason the glucocorticoid receptor is not active is not because it can't bind to a promoter, it's because it's not in the nucleus. So it's sequestered, deliberately kept in the cytoplasm, because its nuclear localization domain has been covered up by HSP90. That strategy is usually not 100%. And depending on the steroid receptor you're looking at, you'll see more or less compliance with that rule, that it starts out in the cytoplasm and upon ligand binding ends up in the nucleus. But it's a general principle that you see for most, with one exception, which we'll see several slides from now. With one exception, they all have this nuclear localization aspect of their regulation. So this would be an example of an activated transcription factor, right? The transcription factor itself is activated. It starts out with no activity, and its activity is brought about by the fact that now it can go to the nucleus and bind to DNA. So how does it really work? Let's get down to where there are colors everywhere. Because I used to think about this in graduate school. What would it be like? You'd have these long discussions on the weekend when there are no movies playing about what if you get really, really small and go inside and you couldn't see anything. You guys going, what were you smoking? I can't say at the moment. You get really, really small and everybody's giggling going, I'm really, really small. Could you see anything? Well, light wouldn't work at that smallness. You couldn't see unless you could bombard everything around you with electrons, which we then did, and we could see everything perfectly well. But they wouldn't have any colors, right? You get really, really small. So this is all a product of imagination, but there's a helix here in the steroid receptor. It's called helix 12. This rotates between two positions. In one position, it covers up the binding site for the next protein, the one that actually has the good stuff, the one that has the hat activity that can start modifying the nucleosomes and activating transcription. The co-activator binding site is covered up by this helix 12. Here you see this case, I believe is estrogen. You see the binding of estrogen in that pocket. And when that happens, helix 12 rotates so it uncovers the binding site. It rotates down, uncovers the binding site for the co-activator that has the hat activity, and that's called P160. So here you see it, helix 12, the co-activator sitting in that pocket. And helix 12 is rotated down onto the steroid sorter, exposing this pocket of binding for the next protein to be recruited, P160, the one that has the hat activity. Some drugs exploit this ability. Let's see how this works here. Tamoxifen, used to fight breast cancer. I think it's sort of an older way at present. But anyway, its binding causes helix 12, instead of to roll out of the way, to make it more accessible to binding P160, it causes that helix to flip the wrong way and make the binding of P160 even less possible. It slams shut that pocket for recruitment of P160. That means the estrogen receptor has no potential to activate a gene anymore. It can't recruit a hat complex. Tamoxifen. Lots of drugs work in these sorts of ways, but this dampens down the whole response to estrogen. Okay, so the nuclear receptor, the estrogen receptor, can't do anything on its own. It has to recruit complexes that contain the chromatin and nucleosome modification complexes. Step one, it recruits a complex that has hat activity. That thing would be called a co-activator, wouldn't it? And that thing is what we just talked about, P160. There it goes, bless its heart. It just recruited P160. Now it can start decondensing the chromatin. But that's not all. In fact, it continues to get on the phone and recruit people to the party. After it recruits 160, 160 in turn recruits other things, other co-activators. Everybody, only requirement is you got to bring your own bottle. Only requirement is you got to bring your own hat. It's not a hatless party. You bring your own hat activity. CBP and a closely related protein, P300, they're sort of interchangeable, that contains a hat. That's a coactivator. PCAF, we've already talked about that. PCAF, the PCAF subunit of the PCAF complex contains a hat activity. So it's hat upon hat upon hat that are being recruited here by this transactivator, the steroid receptor. Step one. Then the coactivators begin to come. So estrogen receptor first recruits CBP, early stages, and later recruits mediator. So how does TBP get here? By association with one of these coactivators. PCAF is traditionally thought to be the one that escorts her to the party. You get these, they remodel the chromatin, and they deliver TBP to the correct address. So you're building the pre-initiation complex, and you're decondensing the chromatin. But this is a series of events. It turns out that P160, as it recruits more and more things, begins to recruit a methylase, which methylates P160. And when that happens, it loses affinity for those other coactivators, but their benefit is already derived. Now, instead of recruiting those coactivators, it recruits mediator. So you see, it's just a cascade of building the pre-initiation complex. It started out with the binding of the estrogen or steroid receptor. It in turn recruited P160, who has its own agenda, first recruiting PCAF and CBP, P300, and then at a subsequent stage being methylated on its arginine, loses affinity for those first coactivators, and ends up recruiting mediator. Oh, I hate these things. Last day too, this is just not right. What do the following complexes have in common? P160, PCAF, P300, they all have a hats on. Yep, still got it. Okay, what's going to follow here is the slide you hate. This will be the one you go back to 10 worst slides. This will have a check mark. There are two major groups of steroid receptors. You knew it would end up in some long list of steroid receptors. You had them in every other class. This kind of list you can kind of look at with squinted eye and sort of get them into the two groups okay. You don't really need, you'll see it's pretty easy to separate them. The first group, these are the ones that you've heard about. Glucocorticoid, mineral corticoid, androgen, progesterone, and estrogen. These bind, their binding site, the element in the enhancer is a palindrome. Two repeats, but they're inverted. An inverted repeat. And glucocorticoid, mineral corticoid, androgen, progesterone, they have exactly the same sequence in that little repeat on each half. Exactly the same sequence. So how do they discriminate among the promoters they should bind? Because they activate different sets of genes if they have the same sequence here. And it turns out that there's one other aspect that you can fiddle with, and that's the spacing of those inverted repeats. And that spacing determines whether it's going to be glucocorticoid or mineral corticoid or progesterone or what's the other one? That's it, androgen. With one exception, estrogen has a little bit of a variation of that sequence, that binding motif here. So what's to know about that first class? Inverted repeat is what they bind, and specificity, for the most part, is determined by how close those inverted repeats are to each other, spacing. That didn't hurt too badly. Now the second group, and it's everything else that sounds crazy. Thyroid, vitamin D, retinoic acid, 9-cis-retinoic acid, RXR. See, already, you could look at that. You don't even have to, you could, across the room, you could say that must be the second group because it's where the crazy ones go, right? Vitamin D3. Vitamin D, it's a steroid. And it has a receptor, steroid receptor, like all the rest of these. The difference here between the first group and the second is severalfold, but one of them is the fact that it's a direct repeat, head to tail. Is that another slide here? Yeah. Direct repeat instead of inverted repeat. Once again, the specificity between all of these in the second group is based on the spacing between those direct or head to tail repeats. What else is strange? The other thing is there is one member here, of course with the strangest name, that binds to all of the other members. They all they bind as heterodimers. So the thyroid receptor really binds as a heterodimer with the other one that's in common to everybody. Of course, that one binds with itself. And which one is that? That's your 9-cis-retinoic acid. That binds with itself, can form a homodimer, and forms a heterodimer with all the other members, the thyroid and vitamin D and retinoic acid. Retinoic acid, of course, is different than 9-cis-retinoic acid. They are the two distinctions between those two groups. There's also one other strange thing about the second group. There's an odd member here, as I say, right there, thyroid receptor. That is the example on the exam and in real life that I'll ask you, I'll say which of the thyroid receptors or nuclear receptors is always in the nucleus, whether or not the hormone is present. There's one that's already pre-bound at the promoter, and that's the thyroid receptor. Of course, it's a heterodimer with the 9-cis-retinoic acid, but nonetheless, the thyroid receptor is always at the promoter, which begs the question, well, are the genes that it binds to regulated in any way? How do they turn on and off? Are they always on? It turns out most of the time they're off. So how does it get to the nucleus and bind and yet no activation? And that's the last part of the story. The SMART co-repressor, SMRT. The default state is when the thyroid receptor, in partnership with the 9-cis-retinoic acid, when they are pre-bound to the nucleus without a ligand being there, without the thyroid hormone being there, the default situation is they have recruited a repressor, a co-repressor, because the SMART complex doesn't recognize DNA directly. It depends on the steroid receptor to do the DNA binding. So in this case, the steroid receptor here acts as a repressor under certain conditions, and under other conditions acts as a transactivator. Because in one case it's complex with coactivators, in another case it's complex with co-repressors. So you see that theme sometimes used, and there are more examples as people unravel the mysteries here of things having a dual purpose. So what are the steps in activation here? How much does it share with all the other members of the family? Well, when the hormone, which is the thyroid hormone, actually finds the receptor and binds it, it causes the receptor to lose affinity for whatever it's bound with. Instead of being bound to HSP90, it's pre-bound to the SMART co-activator. When that change in conformation occurs, the co-activator loses affinity, drifts away, and now you can start the normal steroid recruitment cascade. After that, you can get P160. P160 in turn recruits CBP, P300, and PCAF, and then that's followed by mediator, the regular pathway for gene activation. So that's how steroid hormones or nuclear receptors work in all of us. It's like we took the slide and we tore it apart, and we finally could look out the window and see that we're in a quiet garden, a forest. This is, I told you I would want you to see both the trees and the forest. So here we see the forest, but there are plants in here, as you might suspect. How does it work in plants? There's a 4-hour segment to follow. Just wait. Now, I've found that I'm able to induce genes in mammals. Homo sapien. I can just say plants in the context of this course, and I can see films going over the eyes. That's like a glazing gene. It happens. And people stare, but they still do not see. So this is about as close as I can get to you ever looking at anything that comes after these slides. So I apologize, but here goes. So we're in a lovely garden, and we can smell the jasmine, the sweet jasmine, right? Have you smelled that? Sweet jasmine? It's very strong. That is a plant hormone you're sniffing, whether you know it or not. That is turning on genes in neighboring plants. That hormone is a methyl ester of jasmonic acid. That is a signal, it's a volatile signal that plants communicate that there's a big caterpillar eating me. You should watch out too. So it activates other parts of the same plant and neighboring plants. Everybody's going, ooh, there's caterpillars around here somewhere. Oh yes, the sweet smell of caterpillars. So let's see how that works. There's a rascal up there. Also psyllids. In the genome engineering project, there's a psyllid involved. He's the vector that carries the evil pathogen, Liberobacter asiaticus. And when he wounds, or he or she wounds the plant with its stylet, it triggers the wound response. That's one of the many responses that jasmonic acid controls. The wounding response. When a caterpillar is biting, it's wounding, and that's when the cells are sending out these volatile signals, the jasmonic acid. Also, jasmonic acid is produced in flower development, when the pollen is getting ready to mature. Is that why we think jasmonic acid smells so good? I don't know. It tells us, it alerts us to where trees are that might have fruit, while we still had tails. I don't know. There's no really quantum P-chem explanation for why we think something smells good or bad. It must have an evolutionary origin of some sort, or not. Anyway, so let's look at this jasmonic acid pathway as an example. There are many, many ways genes are activated. And what we're going to talk about just briefly here before we go to the next PowerPoint is this thing right here, topless. My graduate student is working on this. His first talk at the Plant Molecular Cellular Biology annual workshop at Daytona, held at the top floor of some fancy hotel there looking out on the beach. He was the first talk of the morning. It was his first talk, and the title to his slide was A Day at the Beach with the Topless Family. Turns out there's a whole family of these genes in plants, and It woke everybody up. So if you mutate this gene, you guessed it, the plant has no top. It just has roots, which is bad for a plant. So it turns out it's a massive thing. It's got big WD-40 barrel repeats back here, and it's got a tetramerization domain and places where different things bind. We don't understand quite how all of this works, but the student has spent about 8 years trying to figure it out, and we're getting very close. One of the things it does is recruit HDAC complexes. It's a repressor, this thing called topless. Notice that eventually it's connected through a couple of proteins to a transcription factor. This happens to be a transactivator. You don't need to know the name of this, but it's MIC2. It's sitting on a gene, and as long as it's connected to topless, and topless is connected to the HDAC, which would be a co-repressor, then there's no activity in this gene. This gene, in fact, encodes sort of an arsenic compound, glucosinolate enzymes. These are bad news to insects, and so this is normally kept shut down by its association through a series of proteins with this co-repressor that contains an HDAC through this networking protein called topless. So when the insect feeds, it triggers the synthesis of jasmonate, jasmonic acid, the signal, and this activates the glucosinolate enzymes. So how does this work? Insect feeding produces jasmonic acid. That's the little yellow tennis ball. That in turn binds an enzyme that adds polyubiquitin to proteins. And that particular one is specific for this protein here called Jazz. Why do they call it Jazz? Because these molecular biologists in the labs have no life. But you see that? JA, jasmonic acid. People, you didn't see that? And look at this ninja, jasmonic acid. You see what you want to see. Anyway, this is a labile bridge between the co-repressor, topless, ninja, and Jazz. This activated ubiquitin conjugation ligase now comes here, binds to Jazz, and adds polyubiquitin. What is the consequence? You add polyubiquitin to the lysines of a protein. They're targeted now for degradation by the proteasome. So you destroy this linkage. And when this linkage, Jazz, goes away, then this whole co-repressor complex floats away to depress and repress some other gene. And now MIC is released from repression and now begins to recruit as if it was, and it is now, an activator. So you see a common theme here of pre-recruitment, repressors in place, and then with a signal, you change it presto from repression to activation. Synthesis of jasmonate. It binds the receptor ubiquitin ligase. Notice it doesn't, jasmonic acid really, it's a jasmonic acid conjugate with isoleucine. But that's a detail you don't need to know. And then that, of course, binds Jazz, and then that leads to its degradation. Whoops. It goes away, and now it can recruit the pre-initiation complex. Cool. Okay, and that's several other hormones use topless. I won't go into these, but instead of Jazz and Ninja here, this is topless works in several different pathway, hormone pathways. Another one is auxin. And abscisic acid is another wound response signal. In some cases, it recruits MED25, but the detail I want you to know now is EAR, because that's in the Genome Engineering Project, right? You've wondered what this stupid EAR is we keep talking about. Ethylene, what's the other one? Abscisic acid response element, EAR. There's a little sequence within proteins that directly interact with topless. There's an EAR binding pocket right here on topless. So if you have an EAR motif, then Ninja is going to go to topless, and topless is connected to co-repressor, and then Ninja now can connect to Jazz. But the EAR motif is a vital part in this cascade of proteins that bring a co-activator complex to a nucleus. And of course, you can just go to proteins that have EAR motifs and pick it up, design it, and put it in your protein, and now your protein, it doesn't have to be with Jazz and Ninja. You can just stick EAR onto any DNA binding domain, and you have created a synthetic repressor. And that's what you're going to do if you choose to do it in the Genome Engineering Project. You're going to build a synthetic activator, and you're going to build a synthetic repressor, and then you're going to do a hairpin construct if you so choose. Oh man, what's the time? Lots of times? Good. We can go into epigenetics. Are there any questions on this part? How can one gene control all of that? That'll take another 8 years. I don't know, and nobody really does. But of course, you could stop the formation of an apical meristem. And so apical meristem leads to everything else. So it could be very fundamental. So you just, a basic switch, that's probably involved. And you could have no hormone regulation at all. I mean, auxins do the same thing. Auxins have a disposable members called the auxa, the gene that's, the protein that's destroyed by ubiquitination. So it's a common theme. But yeah, how can you, you don't want that in your garden plants. Any other questions? Okay, let's talk a little bit about epigenetics, and I'm not going to try to cover this whole epigenetics lecture. I'm just going to cover it at a relaxed pace, and when we get to the end of the class, we're at the end of the class. The complete lecture is archived from last spring. It's there, covered the entire PowerPoint. The PowerPoint now can be downloaded. And what I'd like to do is, why I'm not going to have a review session, I'm going to spend all day Saturday upgrading the student learning outcomes. I'm going to give you very specific key list of terms, because it's getting to be a little much here. And I want to make it so it's not a bad experience, that you don't just hate me and molecular biology both. I want it to be instructive, informative, but not overly burdensome. So that's why I advise delaying taking the exam maybe till Monday. I'm even considering expanding the deadline maybe to Wednesday, if that helps. Would that help anybody?
Yes.
'Cause I think you probably have lots of exams coming about the same time, right? Okay, so I want to do that then. I'll just go back and tell Proctor U we're going to extend it till, it'll be Thursday morning at 3 a.m., but essentially that's Wednesday night. That'll be good? So that gives me time to open up your student learning objectives, so I can give you lots of specific suggestions on what's important, what to study. Populate the key terms and concepts list so that you have a more directed study experience, as they say.
For the in-person testing, is it still going to be that Monday night then?
It looks like I have to because some people are scheduled and they made plans. And so I will hold it that Monday, and I will hold it Wednesday night also. And the next question is, how about the optional final? Will you be offering the live proctoring then? Because I'd hate to buy a camera for one stupid exam. And yes, I'm going to hold multiple ones. They'll be at night. This is what I signed up for. So I'm going to hold it probably hold it maybe 4 or 5 times and announce those. And it probably won't be a sign-up sheet because there won't be as many people at each individual session, and if there is a line, there never really is too much of a line. I'll just fit you in until everybody's done. So I'm going to be announcing multiple times for the optional final, and I'm going to have the exam 4 deadline extended to the wee hours of Thursday morning, 3 a.m., but we'll have a live proctoring session Wednesday night and a live proctoring session Monday night. And so I would advise holding off on opening when the exam opens tomorrow at noon, or Saturday at noon, because I'd like to spend Saturday, and if I need to, Sunday, but I will make sure I have a very thorough guide for studying so they're not going to flounder and be surprised on the exam. I don't want that to happen here. Okay, so I'm going into the epigenetics PowerPoint, which was, I think I downloaded it this morning. All the days blur, as do the years. Let's go here. Epigenetics. I'd like to get to calico cats. Maybe I will, maybe I won't. Video lectures, that's the archived one you should go to. The PowerPoint is right here, Epigenetics. I've already downloaded it. What am I thinking? Did I close it? I will open it here. The pitter patter of feet leaving. Okay. Yeah, I got a little funky here with Adobe Illustrator. Epigenetics, it needs a musical theme of some sort. It's a study of the changes in the regulation of gene activity and expression that are not dependent on the sequence of bases, but it can be dependent on the modification of bases, methylation being a common example. It's also can be modifications of chromatin proteins like the nucleosomes, the tails of histones. It can also involve regulatory RNAs. So it's a wide range of mechanisms for controlling gene activity, many of which shut gene activity down. Of course, the opposite of that would be de-repression. The next requirement, just by definition, is it must be heritable. And so you'll see many of the things that occur are sort of can walk on either side of the street. There are things that occur in the regulation of control mechanisms inside the life of a cell, and also can be carried on to the cell that is derived after cell division. So what do you mean by heritable? That again is subject to definition. So it depends on the context when you read about epigenetics, how they are defining heritable. Is it through mitosis? Or is it through the life of the organism, to the offspring? Epigenetic changes can be inherited through both of those major transitions. Not all, but some can and some can't. So it has to be a change that doesn't affect the actual base sequence of the DNA, and it has to be heritable at some level. So you'll see examples of things that look just like epigenetic changes, but just by the definitions we choose to use here. And that sort of depends on the context, whether people add this heritability aspect to epigenetics. Originally, it was a very strong part of the definition. So we have to sort of set the stage, first of all, about the big picture of genomes. And how many nucleotides does it take to build an organism? Is there a relationship between a complicated organism like a cat versus a nematode versus a tomato versus a fruit fly? Well, there's just a very rough correlation, but it turns out that Arabidopsis, that small mouse-eared thale that grows in alpine meadows, it has a 6-week lifespan. It grows in a meadow in the high mountains, and so it has to do its whole thing in about 6 weeks, go from seed to germination to flower to seed again before the snows come. Its whole lifespan is very quick. And so it has a very condensed genome. It doesn't have a lot of intergene spaces like most plants do. It has short introns, and yet it has as many genes as any plant. In fact, it has probably a few more genes than we do. So things, how eukaryotes, plants and mammals, vertebrates, have about 25 to 30,000 different genes. So that's what you'll get from this picture. One of the things you'll see, what's a megabase is a million, mega, kilo, 1,000, mega, a million base pairs. So you see the eubacteria, several million, archaea, not so much, but it's wide range. Budding yeast, 12 million. Plants, you go all the way from 63 to giga. That's billions. 19 gigabases in the Norway spruce. How about humans? We got 3. A Norway spruce, you see in those movies where the trees talk and they take great strides through the forest. That's why right there. Get all that DNA. That's got to count for something, right? What you'll see is the genome sometimes becomes endo-duplicated, and it's the same gene, just multiple copies of it. That doesn't necessarily mean more unique genes. Let's look at citrus, 300 maize. So it goes all the way, Arabidopsis, 142, whereas look at Drosophila. Drosophila has less than Arabidopsis, ha ha ha, plants win. Mouse, well, at least we're better than a mouse, right? But look at the pufferfish. Whoa. Oh, that's small, isn't it? Yeah. Yeah, got me. That lungfish, that ancient Lithian crawls into the mud. Look how much DNA it was dragging out. We got rid of most of that to make man. Okay. More fun facts about genomes. Smallest number of it, mean gene size, about 27 nucleotide pairs. Largest gene, over 1 million nucleotide pairs. We studied one of those, remember the Connecticut, Tennessee's ATP Hill or somebody? What usually is asked from here is, what percentage of the DNA of the genome in humans actually codes for protein? 1.5%. The rest of it is introns, and most of it is crazy stuff. The mites and the signs and leftover remnant transposons. 1.5%. If you include all the other highly conserved DNA, and that would be the ribosomal cistrons, don't code for proteins, you only still get up to 3.5. percentage of the DNA in these repetitive elements, which are transposon leftovers, about 50%. But that's an interesting number there. 1.5% of the genome actually ends up as coding a protein. Let's look at this. We've sort of addressed this. This is just a comparison of the amount of DNA, number of genes in different kinds of organisms. You'll see in yeast, 6,000, Drosophila, 14, plants, 25, mouse, 25, humans, 25,000. 25,000, 25,000, 25,000. Drosophila, a little bit shortchanged. Number of chromosomes, those genes can be split up in 4 chromosomes or 23 or more. How about the organization of genes as you go across these different groups of organisms? In the yeast, introns, practically non-existent. There's another thing about yeast genes I found out, didn't know it until last year maybe, or a year before. They have no 5' untranslated region either. They start with ATG. I was a big surprise because I was looking up for the class. They were having trouble. They wanted me to put in the molecular toolbox the Gal4 DNA binding domain. And lo and behold, there was no, and so I began to look. There are only 600 genes, I think, in yeast that have any untranslated region at all. They always start with ATG. So let's look at Arabidopsis plants. They do have introns. The average size is about 2 kb, about the same as yeast. Drosophila, they're getting larger, but notice they're getting larger because the introns are getting larger. And look here at mammals. These things can be 50 kb, but again, it's all because you have repetitive regions. You have an elaboration of these tremendous, long, ungainly introns everywhere. But the actual coding sequences would add up to about the same. This is an overview of what we mean, the different kinds of epigenetic control. Everything is related to gene expression. Histone modifications and variants. Those modifications to the histone tails, there are mechanisms in place that allow it to survive DNA replication, which means they can survive simple mitotic cell division. Transcriptional memory is what it's called. Histone modifications and variants are part of the epigenetic story. DNA methylation, you don't change the base sequence, but you methylate the CPGs. And then RNA interference, and we've talked about this some too, where you get double-stranded RNA, triggers all of these silencing mechanisms with Argonaut, slicer and dicer, and that kind of stuff. They're the three, sort of the triad of epigenetic mechanisms. This is the CPG island, and we've talked about that. Methylation generally in DNA shuts things down, but it shuts things down, you'd think, well, transcription factors can't bind anymore. That's true. That's one way to shut something down, but methylated bases also recruit proteins that just bind methylated bases, and they in turn recruit chromatin, heterochromatin forming proteins. So it sort of works both ways, but generally you methylate the DNA, you are repressing. And it's thought of as sort of a crude, gross level of control, because you can't undo it very, very fast. Roles of methylation: imprinting, X chromosome inactivation, heterochromatin, developmental control tissue, just about everything. You know what imprinting is? That's where you take the marks from the maternal versus paternal parent. There's not many examples of imprinting in humans. That seem there are few, but not very many. This is the CPG islands. Notice it's CPG in 5', 3' direction. Other strand be the same, the C's. Methylation state. embryonic cells are largely unmethylated, and then as you go through development, you begin to shut down whole regions of the genome. So this is one aspect of epigenetics where I think I've talked about it before. We have enough genes to make 100 different tissues and cell types. If you looked at each one of those as a taxonomist or a biologist with a microscope, you'd look through there and you'd think there might be 100 different organisms. They have different transcriptomes, they're expressing different genes, they look different, they have different morphology, and yet it's all the same organism. And so this is a trick that higher organisms have learned. Make it possible to have many variants of your basic life form, of your cell, and just shut down most of your genome epigenetically so that only tiny windows are possible for gene expression. A brain cell, once it becomes a brain cell, it shuts down all the stuff that makes it a toenail. It can never be a toenail. All that stuff is inaccessible to it, even if it expressed the transcription factors that would make a robust, shiny, fungus-free toenails. It wouldn't happen. It wouldn't change. You wouldn't open up your head and find toenails growing there. It shuts down skin cells. The spleen, my favorite organ, the spleen. I'm in a battle with my spleen. Someday it's going to get me, I know. I'm here, I heard every word. I haven't said anything till now. But that's kind of a neat trick. We have the whole genome, all that possibility, and mainly you just shut it down. It's like those jaws in the library across the street there. It used to be great big, the stacks. You'd have to go push a button, and go zing, and you could walk inside. But you kept looking over your shoulder, or somebody at the other end maybe wanted the book in the aisle beside you, and they push theirs and they go, as long as you had weight, as long as gravity worked, there's a pad you can step on that indicates you're in there and you shouldn't be squashed. The jaws of knowledge. So I don't know whether they're still there or not, or whether they're maybe stuck together, I don't know. So, I digress. Okay, islands. The islands are clusters of these CPGs, and they look like they should be heavily methylated. Except what you'll find is a lot of islands are around promoters. And many of the islands are deliberately kept free of methylation as part of the regulatory scheme. When they need to be shut down, then they'll be heavily methylated, but a lot of times they'll be the least methylated part of the genome in these islands because they're usually deliberately placed to control promoters. The pattern of which GPC is methylated, the pattern of methylation is it survives mitosis, positions in the progenitor, 97%. It survives, it can go through mitosis. So the new strand is not methylated, but the DNA methylases are able to bind a hemimethylated site and now make sure the other strand carries the same mark. So that's a pretty easy kind of mechanism to contemplate. How about epigenetic influences of diet? You can read these studies where if your grandma or your grandfather, but not your parents, if they had a poor diet, then you're going to be some way affected. I think height is one of the things. You'll be short or tall or something. Epigenetic influences are not well understood in humans, but they do occur. One easy to understand diet-related is how much S-adenosyl methionine you get, because that's used for methylation directly. So that's a pretty straightforward mechanism, but that's one that's pretty much been worked out. This, of course, is an old diagram, the histone code. We talked about where the tails are, and We've talked about this before. Lysines, acetylated histones, HDACs. We've talked about this. We are not going to need to memorize this at all, but you can see the kinds of things. You get acetylation, methylation, phosphorylation, ubiquitination, sumylation. That usually shuts things down. ADP ribosylation, deamination. All of these different kinds of modifications end up being associated with a particular transcription state. This is where you start to try to make correlations between a specific modification and whether transcription is on or off. And you see some of these can work either way. You see for some of these there's a definite preference to promoter versus non-promoter or towards the end of transcription. And so some of these that are significant here would be acetylation of histone tails on 3 and 4. Acetylation in nucleosomes that are in the promoter region are usually associated with activation. Even methylation, but it has to be trimethylation, not dimethylation. Trimethylation seems to pile up at the promoter and signify gene activation. Here's one that signifies shutdown. It's methylation at lysine 27. What's happening here? This is K4, trimethylation. This would be just generally acetylation, but methylation, trimethylation on K4 is for activation, and 36, you see, can go either way. K9, notice the distribution here. It's active everywhere but the promoter. It's the opposite configuration. Here's 27, where it's definitely associated with inactivation. Ubiquitination of one of the histone 2As is associated with repression, the other with activation. Sumylation is universally associated with repression. The histone, the K4 story, you will need to know, but I will specifically spell it out. But K4 on histone 3 and probably 27, but I will put it in the student learning outcomes specifically, and there'll probably just be 2 here that you need to know. And this is showing the kinds of things that can be regulated. Here's the K4, the K9 story. This K9 is critical. This K27 also has a story behind it, transcription memory silencing. HP1, heterochromatin protein 1 binds to K9 when it's methylated. PC means polycomb complex. This will become more meaningful in just a minute or so. Okay, epigenetic memory. This is the ability to remember the transcriptional activation state before the cell divided. You know, when the cell goes through mitosis, you have to condense the chromosomes down into this horrible tangle, and you get rid of most of the control proteins. And yet you have to remember when you pop out again, you have to remember what you were in your previous life. Now let's see, was I a toenail or a spleen? I hope I get it right. So that's a big deal, having to go through chromosome condensation where essentially all gene expression shuts down. You get rid of transactivators and pre-initiation complexes and all that stuff, and you have to build it all back again just like it was. Transcriptional memory. So we know a lot about that by studying 2 big complexes of proteins which are antagonistic. One works to shut things down, and the other works to keep things active, genes active. And these 2 combatants or polycomb versus trithorax. These, what crazy names. These were all studied and first characterized in Drosophila, in insects. And polycomb refers to the sex combs. If you have polycomb, you have the sex combs popping out at crazy places. That's how it was named. You have mutations in polycomb where they said, look, this guy has got all these sex combs. We'll call this gene multiple combs, polycomb. Then 20 years later, they figure out how it works. Trithorax is involved in the developmental process that builds a thorax, segmentation. So we'll look at these two, and again, this is a lot of information. So I want you to just kind of sit back and get the big picture here, and I will identify specific things you're going to need to learn. So this shouldn't be squinty, furrowed brow. This should be tell me a story. So here goes. How does this polycomb thing work? There are several models, and we don't know completely because, as you know, we don't really know what euchromatin and heterochromatin looks like until, last summer. But one mechanism is that it globs together, it has affinity for itself, and it holds a highly condensed form of the chromosome. It stabilizes it so that it's in a sort of a heterochromatin configuration by just having attraction to itself and to specific histone marks. It's condensed, polymerase, activators, nothing has access. An opposing theory is it doesn't cause things to condense down. It simply directly interacts with all the important players. It masks, it gets in the way of building the pre-initiation complex. So here's some more of the same. It's supposed to, with mutual affinity and with affinity to specific marks, just start building on itself, making these creeping domains that expand and expand. Another is that it's attracted uniquely to promoter sequences, that there are proteins here in promoters that have affinity for polycomb. So that's not very informative. That's the kind of simplistic models, nice color, kind of pastel, kind of pleasing to the eye. Didn't really tell you much. We're going to get some more nice shapes and floating globs because we don't know much more about it. But we do know that with polycomb there are two major subcomplexes. Polycomb group, polycomb complex 1 and 2. And they have different enzymatic activities and binding properties. So let's see how this works. PRC1 is, or polycomb repressive complex 1 is the first, and then PRC2 is the second. The first, and it's with this sort of purple slate gray shape, it binds to H327 trimethyl. So that now we begin to understand why that's an important mark, because we know a lot about what it attracts, what binds there. Polycomb, PRC1 binds there. And it's thought through the previous slides that just simply binding it, it has affinity for itself, it begins to bunch the DNA together. So does that fit in with the new idea of the higher orders of chromatin being just figments? It does, because you can cluster things together in tight masses without having any kind of higher elaborate folding order. And when we look at it, we see these little knots of proteins that have affinity for each other. So it would work in the present model also. Notice it also catalyzes, it has enzymatic activity, it catalyzes the ubiquitination of lysine 118 of H2A. This lysine here is actually in the body of the histone, not on the histone tail. And we'll see up here that actually, we don't quite know how, but it seems to inhibit the phosphorylation of serine 5 on RNA polymerase, the CTD. So where it can directly control or shut down transcription, because you can't, if you can't put that first serine 5 phosphorus group on, you can't get out of initiation mode. So let's look at the other one here. It also binds the same thing as PRC1. And it catalyzes the enzymatic activity, the histone methyltransferase. In fact, it binds to the same thing that it creates. So if you can just get it started, it binds here and then flops around in any neighboring nucleosomes. It will put the same mark that its other domain is binding to. So it creates a domain for a second PCRT to come, and then that's how polycomb creeps. So, and creating these this histone 3, lysine 27 trimethyl modification prepares the way for binding of more PRC2 and PRC1. So you see how the thing, once it gets started, begins to grow. Shut down. Okay. And in the other corner of the ring, trithorax. It has a similar kind of activity. It has subcomplexes. The first is called ASH1, contains a methylase domain, a set domain is methylase, but it is now methylating lysine 36. It has something called trithorax C terminus, and it has an N terminus, and they do different things. The set domain here methylates K4, and trithorax N terminus stimulates our friend CBP, mouse coactivator, to acetylate 27. Remember, polycomb methylates K27. So it's a battle to see what kind of modification should be placed on histone 3 tail, lysine position 27. And then it has bromodomains, and they bind acetylated lysines and phosphorylates RNA polymerase CTD at serine 2. See how that works antagonistically against polycomb? Polycomb is trying to prevent the phosphorylation of the CTD. Trithorax promotes phosphorylation of the CTD. Remember, this is the second one that comes around, right? This alleviates the pause, serine 2. So, how does this survive replication? How do you get, when you decondense after your cells have divided, and now you have to repopulate your chromatin with the correct transcriptional control, how do you do this? And the first thing you're going to do is make sure polycomb and trithorax are distributed in the same regions they were before mitosis. And so the theory is that the marked nucleosomes in the pre-replication DNA, these marked nucleosomes are randomly distributed among the two strands that are made after the duplication event. So you don't get all the old nucleosomes going on to one of the daughter genomes versus the other. And because, in effect, that seeds the newly made chromatin with the marks of the previous generation. And these complexes, whether it's trithorax or polycomb, are able to bind and then spread the mark specifically for them. So that's how they do it. Of course, these proteins, both trithorax and polycomb group, have affinity for single-strand DNA. You know, they are binding to nucleosome tails. There's no single-stranded DNA, for the most part, on a nucleosome. So why would they want to have single-strand binding affinity? It's for this particular reason. So they will be attracted and have affinity for brand new DNA as it's being replicated. That's just a brief moment where it's single-stranded. These are going to be following a replication fork, hovering around like bees. I won't go through the details here. Right, so it survives it through this event. Let's see what this is saying. When the chromosomes condense, where everybody has to leave. Okay, we're shutting down, everybody leave, turn the lights, they blink, last call, everybody walks out the door. They throw the furniture and everything out in the yard, except a few essential pieces. They let trithorax remain. That's for activation. They throw away polycomb, all those ruffians who just want to shut things down. So that's something that we don't quite understand. Trithorax remains. Just a little fun fact. Let's look here at another concept. This is a concept that will be on the exam. And it's called the methylphosphate switch. And it involves the turning on and off of a particular mark with another mark. You remember lysine 4, lysine 9, 27. If you look at 4, 9, and 27, what you will see is right next to them, there's an amino acid that can be phosphorylated. In the case of lysine 9, it's a serine. In the case of 27, it's a serine. These are definitely known to act as switches. If you have a phosphorus group here, you destroy the ability of 9 to bind something. So in the case of 4, it's postulated to be a switch because instead of a serine, it has a threonine that can be phosphorylated just upstream of it. Okay. Next mechanism. We finished histone modification. There's a lot of information there. I'm going to condense it down to a few principles so your heads don't explode. Next thing. Here's a surprise. Non-coding RNA. It turns out most of the genome, even the stupid stuff, gets transcribed. RNA polymerase will go through it. And many stretches that don't seem to code for anything will end up as a transcript. It'll be low abundance. And so people go, aha, I knew there was something there. We keep looking through this. We all called it junk. How wrong you were. In future generations, you'll know it was not junk. Maybe it's junk. But some of it is not junk. Maybe it's junk that's, we got all this junk laying around here, might as well, I can see and look through, I can use that little piece there. Some of it is now being used for regulation. I don't think all of it is. I think most of it is junk. But there are specific examples where regulatory schemes have very deliberately taken advantage of long stretches of non-coding RNA. Here's RNA. It's used structurally for ribosome scaffold. It's used to code the proteins. It's used for tRNAs. And now it's being used as a mechanism of gene control itself. See what it says. Only 1 or 2%, we know now, it's 1.5%, mammalian genome encodes proteins. 70 to 90% of the entire useless genome is transcribed at one time or another. Human mouse transcriptomes contain 80,000 to 180,000 distinct RNAs. Now, how many genes are there? 25,000. And yet we have distinct RNAs ranging from 80 to 180 different species. These transcripts range, of course, from 50 kilobases to 100. So it's thought and been demonstrated that particular specific hairpins in non-coding RNA actually are recognized by polycomb. Polycomb is on the search not only for chromatin to modify, but for RNA. Now just think of this. If you have a long transcript, 100,000 nucleotides, a long string of baby gators, and who are the baby gators? It's polycomb. They're riding and they're floating around, and when they land, you have now tethered a heterochromatin forming complex here, and tied to that is another one that's kind of bouncing around, and once one binds, then the greater probability that a second will bind. This is synergism. It also means that if you have a polymerase making a long transcript, it would be possible to make sure that polycomb, if you wanted to shut down this region on either side of you, that polycomb is not going to float away and start repressing places of the genome that should not be repressed. It's going to tether, it's going to make it so you can shut down your X chromosome and leave the other one untouched. Because you've got all the baby gators holding on to the rope. They can't go to the other X chromosome because they're all tethered together with this long hairpin-filled non-coding RNA. And that is apparently what happens on X chromosome inactivation in females. So here you see both the PRC1 and 2 have affinity for RNA domains, these hairpins. Right, and of course these mark it for repression. It's used in mammalian X chromosome inactivation and plants. When they produce a flower, flowering, that's always a hard word for me to say, flowering. Flowering. The more you say it, the more you can't say it. Flowering, yeah, I can't say it. It's something you want to tightly control. You don't want flowers busting out at the tips of your roots, do you? That would be bad. That's just like, where are all the flowers? You got to dig for them. So you keep that tightly controlled. Only when the plant is full and produced lots of sugars and storage proteins and nutrients, and it's ready now to make fruit and multiply, do you want the flowers to pop out. And its regulation is controlled by non-coding RNA and something that looks like polycomb, even in plants. Oh, there go the eyes. What's that? Oh, there's another one. That's chromatin modification happening right here. Okay. Let's talk about the female problem. That extra X chromosome. it's never expressed, right? It's there, but only one at a time. The males have as many active X chromosomes as females do. In the female, though, a choice is made as to which one will be active. And every cell that derives from that one cell that made the choice has that same pattern. It's inherited. And so you'll get, depending on when that decision as to which X chromosome to inactivate, you'll get a chimera of cells. All cells derive from the one that made the decision. And that's what you see in the calico cat. So you see this only in the female because you see two patterns at the same time, the orange tabby and the black. Each is carried on a different X chromosome and depends on which one you randomly shut down as to which coat pattern will be displayed. So that big patch of orange, they all came from that one cell that decided that the orange tabby X chromosome should be active. In other animals, it doesn't occur in skin coloration or hair pattern. The decision as to which X chromosome to inactivate occurs at different times. But you see, in the organism as a whole, both are expressed. It's just in a particular cell, only one is being expressed at a time. So if you looked under the microscope and you stained for DNA using something like DAPI, you'd actually be able to see a region that if you looked long enough and this was your full-time job, you would see a thicker region near the side called a bar body. And that's how you can look at a cell and tell whether it's a male or female, just at the cellular level, because this is the inactivated, condensed X chromosome. So this is how much time do we have? 10 minutes?
No, 15.
I'm sorry.
15.
15? Okay, and take a time here. Okay. This is the main switch right here. These two things. And they don't really code for any protein. They both code for non-coding RNA. And depends on which wins the battle as to whether that particular X will be on or off. So let's see how this works. The first gene going in the left to right orientation is called ZIST. And it has really two promoters. This one's expressed first, and then the second one's is expressed, and you get all of this going. Going in the opposite direction is one called T6. And we don't worry about the two there, but it has kind of a two thing going on here also. But the bottom line is it's sort of the antisense of ZIST. So you're going, what a crazy name. T6 ZIST? What does that say backwards? Yeah. There's molecular biologists. They don't watch enough TV. They got nothing. I mean, they just sit up thinking up these kinds of things. ZIST and T6. And these are the ones we're going to concentrate on, the silencing. These are just non-coding. So ZIST is expressed in inactive. If this one wins, ZIST, you get permanent shutdown. If T6 is active, you get an active X. So how does this work? So the T6 is green and the ZIST RNA. In the predetermination cells where both of them are active, you have transcripts coming from both, and then you get the extinguishing of one or the other. In this case, T6 is extinguished. T6 being the green ones is extinguished on one, but keeps going on the other eventually. So first thing to happen is one of them loses the ability to make T6, and it gets coated with, what's it being coated with? It's being coated with that non-coding RNA that comes from ZIST. That ZIS gene, that little red ZIS gene, is making lots and lots of copies of itself, and that RNA is crawling along the chromosome, the X chromosome, and shutting it down and completely coating the X chromosome that is doomed to be shut down. And that's the beauty of the non-coding RNA, the fact that it's produced, it binds polycomb, and then it sticks right to itself. And now that you've stuck it to itself, it can't float away and inactivate the other X chromosome. And that's what keeps it localized to one of the X's. So it just has to start on one, and then that one now is doomed because you'll continue to make more and more and more, and they'll just bind and unbind and crawl along like a creepy crawler, but always attached, always tethered to the original X that one of their members bound to. So that's this kind of the overall strategy. Both express high T6 transcript, very low ZIS. One of the T6 promoters recruits all of the transactivators. The X will become active and the other one will be shut down. So what's happening here? One of these, one of the two T6s gets all of the transactivators. How does that happen? It turns out the transactivators are sticky like bubble gum. And there's a step here where the X chromosomes come together. Like boom. Well, now they're great lumbering cables and they come together. But each one's got a sticky glob of bubble gum on it. The OCT1 and the CTF, these transcription factors that are at the T6 promoter. But it turns out these two bubble gum, these transcription factors, have greater affinity for themselves than for the DNA they are bound to. So when they get together and then they separate, the bubble gum goes with the bubble gum. And it's just a random series of events as to where all of the bubble gum will end up. It can't go to both because the transcription factors have great affinity for themselves, more so than the promoter they're bound to. And that is the molecular decision. That's a binary switch that controls the fate of that particular chromosome. So in the one that is not making T6 anymore, that means all you're going to make is ZIS, which is pumping out the non-coding RNA, which is attracting polycomb, which is growing and growing and spreading and coding the whole thing. This one here, that now is just making T6 alone. T6 is the antisense of the other one, so it keeps ZIS shut down and constantly keeps it open because ZIS can never be active again. So let's see that illustrated a little clearer here. Here you see the transcription factors. The one you're going to need to know is the CTCF. You know, I've been reading about this. It's a dangerous thing. But CTCF, that's that invention that metazoans have. That's the looping protein. That binds cohesin to make great loops. I'll have to ponder what that means later, but this is one of the sticky proteins that goes on one or the other, but not both. So all of the transcription factors, first, both the T6 promoters are active, both have transcription factors, CTCF, and I think that's OCT4 or something. Well, it's not on here, so it's not important. But then the two pair, and they make contact, and it goes to one or the other, but not both. A symmetry break results in one now is able to activate the ZIST, and T6 on this one is forever wide open. So let's look at another. This loss is able to express the rep A transcript. Rep A is, you remember that first little promoter? Cyst has two. The first one is called rep A. And that's where you have a clustering of these hairpins that bind PRC2. So it turns out that the first promoter is active, but the second promoter, that does all the work making these long non-coding RNAs, that one requires that the first gene be on first before it can be on. So you go, that seems way too complicated, right? There's some of you are still awake. You're drinking too much Diet Coke, I think. You should be totally asleep by now. It turns out, believe it or not, this is a little bit interesting, that this PCR2 is recruited and it actually binds right around this region on this little short non-coding RNA. It turns out that the strong Zis promoter actually requires heterochromatin. This is crazy. This promoter needs to be embedded in heterochromatin before it has any activity. That'll take some more reading. That'll be like 4 more PowerPoints for the summer. I'm sorry. So that's essentially the story there.
Dr.
How much are we going to need on exam? Yes.
How does the PRC2 bind if it's an heterochromatin?
Well, it binds to nucleosomes because so it binds and then spreads. And so it's populating the area around the promoter of Zist first. And then as Zist starts making transcripts, these things float off. They stick nearby because this is a very long transcript. So it just flops around and binds other places. And once it binds, the thing can kind of migrate as one gets off and the other one gets on. It can kind of walk along in different directions and it ends up just crawling over the entire X chromosome and shutting it down. But this promoter has to be very special because eventually this is going to be nothing but heterochromatins. So you want to make sure that promoter knows how to operate in heterochromatin conditions. And that's still somewhat of a mystery, but some of its transcription factors have affinity for heterochromatin protein 1. So you just have to keep a small enough region around the promoter open all the time. 5 minutes. Okay. RNA-based epigenetic mechanisms. That's the third leg of the triad of defense. Oh no. So we're going to try to make this painless, if that's possible. You've heard the name just thrown around without much regard to definition. MicroRNAs, hairpin RNAs, they're not the same thing. MicroRNAs have a very distinct secondary structure. Hairpins with bulges and hairpins that a specific complex of proteins has evolved to recognize. And it's a whole unique processing pathway, a microRNA. A hairpin can form any place, and it has another processing pathway. But microRNAs, they have their own names and stuff, have a strong connection with regulation of gene activity. If you get a microRNA on your case, it means shutdown usually. But usually one can't do it. You'll have to get several microRNAs binding. So where are they found? They are found most any place in regular, they are found in a messenger RNA. Here's in a pre-mRNA. They are found in non-coding RNAs. They are found in 3' regions. But notice all of these things have poly-A tails. What does that signify? It means the transcripts that carry these microRNA, very specifically structured hairpins, used to be messengers. Or at least if we can't go that far, they at least are the product of RNA polymerase II. We can say that for sure. That's the only enzyme that would put a poly A on the end because it's dragging along this CTD that has that complex. So they all are polyadenylated, so they're made by Pol II. Some of them, they have introns and stuff. Many of them could have been messengers at one time in their history. The primary RNAs always have 5-methyl caps. Another hallmark of being made by Pol II. Okay, the microRNAs first. You get it made by Pol II, you get it processed out. So the hairpin is preserved and everything on either side is cut out. And that's done with a protein called DRASHA. DRASHA in animals, but not in plants. DISA does this in plants. It's exported to the cytoplasm, and then you get DISA. DISA now cuts it up into smaller parts and eventually delivers it to argonaute, which takes it to the target RNA for silencing. Single strand, and this is an RNase, and the RNase argonaute uses a guide RNA. When that guide RNA is perfectly aligned to a messenger RNA, it now cuts. That's what we used to think. It does that in plants. But the main mechanism of shutdown in animals is a little different. So in animals, most silencing show only 6 to 8 nucleotide sequence homology to the target site. So in animals, if you're doing genetic engineering, this has wide off-target effects. But in plants, it's much tighter. The entire 21-mer is used for homology. And actually, the Argo actually makes a cut. In animals, it's a little bit different. This silencing complex binds at multiple positions on the 3' untranslated portion of the message. Why is that? And it's related to its mechanism. In plants, Argo makes a much larger homology base pairing and can cut any old place. And it silences gene expression by just cutting and destroying the RNA. But in animals, you'll notice there hasn't been much cutting going on here. Here you see, in plants, it leads to cutting. This is how it works in us. Did you know that when your RNA is exported to the cytoplasm for translation on ribosomes, that it's a big horseshoe? The 5' cap, the methyl cap, is actually bent around and making contact with the poly A tail, like a dog chasing its tail. This is how silencing works here. Argo doesn't cut, but it attracts a protein called GW182. GW182 has affinity for, or attracts the adenylase complex. When it recruits the poly A deadenylase, it starts destroying the tail, which reduces the half-life of the messenger RNA. In addition, this shortens the tail. The deadenylase complex also gets in the way of the cap region because it's all here right together. It interferes with ribosomes loading here at the cap. So you've got termination and deadenylase happening right where you should be starting translation. So it interferes directly with translation. It doesn't interfere by destroying the messenger RNA. And that was sort of a surprise. And this you never know, but I went as this to Halloween. I don't think anybody got it. They said they did. Well, yeah, we got it. So this is what I did. I had a red cardboard thing. I went to the hardware store. Lowe's gives you inspiration, see? And I looked at WD-40. You know, it's got the red cap, it's got the red straw, and the big shield that says WD-40. I went, yeah, I can make a spear out of that. I got cardboard, I had a spear and my cardboard cap, a skull face, of course, and I went as the penetrator, probing the heterochromatin, of course, looking for chinks in the armor. Nobody really got it. So in plants, I told you there were two rogue polymerases. They have disguised themselves as Pol II. They try to get into the heterochromatin. The heterochromatin is, you know, the inherent part of the dementors that control the big prison, I've forgotten the name of it, that place that nobody, Abscam or someplace you don't want to go. So these guys are dementors, these two polymerases that are probing for chinks and who's going to get out, you know. And I had my spear. And so these two polymerases are dressed up like Pol II, because if Pol II can get in there, I mean, if these two guys can get in there and transcribe, then Pol II can get in there. And what's the danger there? That Pol II gets in and starts reading transposons. Genome instability. These are things you want to be permanently shut down, and you have these polymerases that are asking the four questions. So the first one, Pol IV, goes along ahead of time. It doesn't look for a promoter. It can, if it gets a way in there, it can start transcribing. And so what you'll see is that it is looking for, it's paired up with this protein here, SHH1, which is looking for this histone mark that says, I am heterochromatin. Because these two ruffians here, Pol IV and 5, you don't want them roaming free throughout the good stuff. You want them just to be climbing over the heterochromatin, looking for chinks, people trying to escape, genes trying to get transcribed. So the first one goes along and says, am I in heterochromatin? Yes, because you have read H3K9 methylation. You're in heterochromatin, so it goes fine. Now, is there a chink? Can I get in here? And if it can, it makes a short transcript. And this transcript, in turn, is used by a second RNA polymerase, but this polymerase uses RNA instead of DNA as a template. This thing makes double-stranded RNA for silencing. And this thing goes up here and is processed by dicer and the 24mers, and the ends are methylated, and then it binds a special argonaute. And this argonaute takes that message and now goes to the other Thug2, the second polymerase following along, master and blaster. This guy has a little short hamster tail. Not a full long like the queen of transcription, Pol II, that drags along. This is a little GW motif that this argonaute binds. And it brings the guide RNA to confirm that really it has found Pol V, Thug2. And once that gets there, that argonaute recruits a methylase, which methylates the DNA to make sure it's totally shut down again, locks the gate. And what keeps Pol IV from just roaming into the good genes? It is interrogating for SUV. It's looking for this protein, which in turn binds to previously methylated DNA. So one is saying, do I have the right nucleosome chromatin mark to be in heterochromatin? And the second one is saying, am I in a region of DNA that's heavily methylated? And so you see these double team, these two guys, they're very closely related to Pol II, but they are different because they are just masquerading. And that's the end of the tale. Whew. Oh man, you got your full tuition. And so this is way too much to learn, gone over way too fast. So I will write student learning outcomes and make it so that it can be learned in a reasonable length of time where you get the main points. I just wanted you to know the trees are also very interesting.